Hello, everyone. Welcome again to another virtual Zoom lipid signaling seminar. This is the last of this format. We will be back in a couple of weeks. So a couple of announcements. We are reforming under the auspices of the Lipid Research Division at ASBMB. We really want to thank George Carman and Rob Stalen, who are the head of the LRD, for their support. And so essentially the format will change a little bit. We're probably planning to change the time and date to make it a little bit more convenient for people from Europe. So it'll probably be moving a couple hours earlier in the day. And keep an eye out on your email. We'll probably send an email out just with a poll on what time and date people would possibly prefer. Another announcement, we're going to have a couple new people joining us and helping to organize this. So Amy Kiger, Julie Brill, and Ray Blind will be joining me and Mike in helping to organize talks. And so you'll see their faces in the upcoming weeks. With that, I'll hand it over to Mike and he'll do the introduction for today. And thanks again, everyone, for your support. Mike and I have really enjoyed running this seminar series.
Thanks, John. So I'm going to introduce Makaia. She's been working in the lab of Yorgo Skiniotis, who had a scheduling conflict come up last minute. So you guys have to settle for me. So Makaia got her PhD in 2017 at the University of Rochester, where she worked on the folding of G proteins in the laboratories of Gregory Tall and Robert Freeman. She did a short postdoc at Rochester, and then very recently in 2019, she moved to Stanford in the Skiniotis lab, and she's done fantastically well during her PhD and her postdoc, being awarded foundation fellowships and some very nice papers that you're going to hear about today. So with that, I'm going to hand it over to Makaia to give her presentation.
Okay. Thank you for the introduction, and thank you for inviting me to talk about my research. I really appreciate the opportunity. So just starting off, just so we're on the same page, neurotransmission heavily relies on neurotransmitters in the nervous system that are released from presynaptic to nerve terminals, either from inhibitory neurons or excitatory neurons. They're released into the synapse and then bind at the postsynaptic cell, either at ionotropic receptors or metabotropic receptors. And these metabotropic receptors are G protein coupled receptors, or GPCRs. GPCRs are 7-pass transmembrane receptors that when they bind to a ligand, engage a heterotrimeric G protein. This stimulates the G alpha of the heterotrimer to release GDP and subsequently bind GTP, allowing for a functional dissociation of G alpha and G beta gamma to then go act on their respective effector enzymes. Now we can classify GPCRs based on their sequence and structural homology. Class A, or rhodopsin-like class of receptors, is the most historically well studied. It is more of a structurally the simpler class. We then have class B receptors, which are peptide binding receptors, and these can bind either exogenous ligands or internal peptides ligands. You can also have class C receptors or class F receptors. And today I'm going to be focusing primarily on class C receptors, as that's what GABA-B is. This class of receptors is characterized by having these large extracellular domains called Venus flytrap domains, and having a linker region. And then another main characteristic of this class that other classes don't have is that it's a dimeric receptor. So these class C receptors exclusively function as dimers. And so the focus of my talk today is GABA-B. And GABA-B binds the inhibitory neurotransmitter GABA. And when it binds GABA, it activates the GIO class GIO class of heterotrimeric G proteins, which allows for the inhibition of adenylyl cyclase and the activation of G protein inward rectifying potassium channels to cause an efflux of potassium. And when it inhibits adenylyl cyclase, it's causing an inhibition of cyclic AMP production. And these two things allow for a neuronal inhibition. Now, I'll also mention a couple of times mGlu5 as another class C receptor that I'm comparing to. And just so you're a little more familiar with mGlu5, it binds the excitatory neurotransmitter glutamate and then activates GQ signaling, and downstream that leads to an increase in intracellular calcium and neuronal excitability. Among the class C receptors, GABA-B is really unique for a number of reasons. Now, all other class Cs, and here I'm saying this is mGlu5 as an example cartoon, but mGlu5 is very similar to the rest of the class C receptors in that you have your venous flytrap domain, a linker region, and a transmembrane domain. What's different about all the other class C receptors versus GABA-B, though, is that this linker region with other receptors has a cysteine-rich domain here. And we had actually found in a previous study with M5 receptor, structural studies of the mGlu5 receptor, that when you activate mGlu5, it's actually this cysteine-rich domain here is crucially important for transducing the signal from the venous flytrap domains to the transmembrane region. Now, GABA-B does not have a cysteine-rich domain. So before obtaining the structure, it was unclear how the signal was being transduced from the venous flytrap domains to the transmembrane region. Beyond that, also, a unique aspect of GABA-B is that GABA-B is an obligate heterodimer. And this is because GABA-B1 is able to bind ligand, but it cannot engage G protein. And GABA-B2 is not able to bind ligand, but does engage the G protein. It's necessary transactivation between GABA-B1 venous flytrap to the GABA-B2 transmembrane domains. So in our lab, the primary way we go about studying these G protein coupled receptors is through cryo-electron microscopy. And basically doing cryo-EM, we pure protein to be a homogeneous sample, and we then freeze it in vitreous ice. We pass electrons through our sample, and then from that, we're able to get images of our individual particles. We then select for the individual particles, and then we back project these particles to make a reconstruction of our protein. And our lab has done this pretty successfully with a number of GPCRs in the class A receptor with the respective G proteins, class A receptor with beta-arrestin, which is a protein involved in desensitization of GPCRs, class B receptors, and a couple of years ago, we were able also to get the mGlu5 receptor and look at the class Cs as well. So given the unique way that GABA-B is structured, or the sequence of GABA-B, we want to see how does the structure of GABA-B differ from the other class C receptor that we have a structure of. So here is the cryo-EM map that we obtained of GABA-B, and here in teal is GABA-B1. You can see the ligand is bound here in gold, and we have GABA-B2 here in tan. There's also these, I'll point out in this map, so this is a GDN micelle, or GDN and CHS micelle. That's what the purification was done in. And these here are actually not part of the GABA-B receptor. We found actually that there was structuring in the micelle at that region, and we believe these would be either GDN or CHS, that they would fit these ring structures of either GDN or CHS. We're not confident in saying which one each of these is. So we found a number of interesting things when we analyze the structure of the receptor. For one, we have this ECL2. So the cysteine-rich domain in the M-glue structure and class C receptors, other class C receptors in general that have the cysteine-rich domain is very important for signal transduction. When you have the apostate structure, you can see that the two protomers of mGlu5 are separated, and then once activation occurs, you have kind of a twisting around of the receptors. You have a closing of the VFT domain, and these cysteine-rich domains then can interact, and you bring the two 7TM domains closer together at a different orientation. We found that the ECL2 loop actually compensates in GABA-B. GABA-B, the ECL2 loops compensate for not having a cysteine-rich domain, and they form this beta sheet structure along with the linker region in both GABA-B1 and GABA-B2. We also recognize that compared to other class C receptors, the interface between the two protomers in the inactive state was much closer together with GABA-B than it was in the mGlu5 structure we had determined earlier. In fact, there's even an action at the intracellular side of the receptor between histidine and glutamate residues. Here you can see if we turn the receptor, so we're looking from the intracellular side up, you can see how the histidine and glutamate residues between the TM5 helices and TM3 helices are coordinated. And if we mutate these residues, we actually have an increase in constitutive activity, suggesting that these residues are actually forming an ionic lock, inhibitory ionic lock when it's the receptors in the inactive state. And then the most striking and surprising observation we came to when we were studying or looking at the structure of mGlu is that we had phospholipids bound actually within the core of the receptor, the 7TM cores of the receptor. So if we look from a top-down view, you can see it's right in the middle of these 7TM helices in both the GABA-B1 and the GABA-B2 receptor. Now, this is really interesting for a number of reasons, but one of the things that's different is different about the sequence of GABA-B1 or GABA-B in general is if you compare to other GPCRs have this something called a toggle switch motif, which includes a tryptophan residue that faces in towards the middle of the 7TM bundle. And during activation of the receptor, this toggle switch tryptophan actually changes its orientation during activation. Now, GABA-B receptors actually don't have a tryptophan residue there, and it's actually very important to allow the binding of this phospholipid. Instead of having a tryptophan, they have a cysteine residue in the same, and that allows for the space for this phospholipid to bind. So looking at the cutaway maps here so that we can look at the densities of our phospholipid that's bound in the core here, you'll see it's a wishbone shaped like density. We have one tail of phospholipid going in core. We have another tail that kind of kicks out in between TM5 and TM6, and this is true in both the receptors as far as the general orientation of the lipid. Based on the densities, we had better density for the GABA-B1 versus the GABA-B2 phospholipid. So when we were modeling in or thinking about what phospholipid this could be, we used the GABA-B1 receptor map. By process of thinking about what lipids are available in insect cells, so the main lipids that you would phospholipids you would find in insect cells would be cardiolipin, phosphorethanolamine, phosphatidylcholine, or phospholinositol. Cardiolipin has more than two tails, so that one we were able to toss out. Then we looked at and we docked in using GemSpot, which our lab in collaboration with Schrodinger has developed, and that's a program or a program that allows you to dock in different ligands into EM densities. And when we use that on phosphorethanolamine, phosphatidylcholine, and phospholinositol, we found that phosphorethanolamine fit best into our map. And we also looking at the region and residues that were creating the environment for the phospholipid head group. Phosphethanolamine made the most sense. You can see here phosphatidyl is just a little bit too big for the space, and phosphatidylcholine, we didn't see, at least in GABA-B1, that the right residues would create the environment to allow for that to bind. So we modeled in phosphethanolamine into GABA-B1, and that we were fairly confident with GABA-B1, that's what it was. And for GABA-B2, we modeled phosphethanolamine in, although we were a little less confident with that one. And I'll say that there's another paper that came out in the same issue as our paper. Had come out on GABA-B, another structure of GABA-B, and they actually were able to do mass spec on the lipids, and they've had 50% occupancy of, or I shouldn't say 50%, they had 50% of their lipid they found was phosphethanolamine, and 50% was phosphatidylcholine. And their interpretation is that the GABA-B1 lipid is phosphethanolamine and the GABA-B2 lipid is phosphatidylcholine. So we want to see what the consequence was of having this lipid bound to the receptor. So we used a functional assay in which we have a chimeric G protein that has a C terminus of GI that allows it to bind GABA-B, but the rest of the G protein is a GQ, which allows signaling through PLC and allows for the accumulation of the downstream metabolite IP1. So we can use this assay to test the activity of our receptors. And if we compare to our wild type, we made 2 types of mutations to the GABA-B receptor to probe for what the lipid was doing. And the first type of mutation we made was trying to exclude the lipid. So we mutated lysine residues to tryptophans to try and displace the tails of the lipids. And we did that, we actually saw that there was an increase in efficacy of the receptor when you treat with GABA. And the other type of mutation we made, we looked at the head group and the residues that would be interacting with it. And this arginine residue, here's a top-down view. This arginine residue interacts with oxygen here. So we mutated that arginine to an alanine. And when we did that, we also saw an increase in efficacy, although we also saw an increase in the potency when you treat with GABA. In addition to that, we can also look at the basal or constitutive activity when we're not treating with GABA. And so this assay here on the line, you can see this line here is wild type receptor, and at different increasing concentrations of transfected wild type GABA-B. In blue is our arginine mutant, and in purple is the mutant that would displace the lipid tails. And you can see that the constitutive activity is actually lower for the mutant that displaces the tails and higher for the mutant that changes the coordination of that head group. Now, this is really interesting that it has some implications for how this lipid could be sensing, signaling, and transducing the signal within GABA-B. We also conducted the look at this, the importance of this lipid. We used molecular dynamics simulations in which we started simulations either as wild type, like as our structure shows with the lipids bound in the core, or we started simulations in which we had removed the lipid that was in the core. And allowed the simulations to progress for 200 nanoseconds. And you see fairly rapidly if we don't have the lipid and we measure the 7-TM cavity volume, you can see there's a decrease in cavity volume that happens quite rapidly. So you actually have the 7-TMs collapsing in. And if we look at this in data as an ensemble values, you can see there's about 30% decrease in the cavity TM, 7-TM cavity volume. Interestingly, we had some simulations in which lipid surrounding the receptors actually invaded the 7-TM domain. And it invaded at the same spot as where we saw that one lipid tail kicking out between the TM5 and TM6. It would actually invade in the same position. And when it did invade and we had external lipid entry, you can see that we did not see a decrease in cavity volume. Here you can see a number of simulations in which we have the lipid invading from the outside. And here we have 2 tails of lipid coming in this simulation. And if we extend this simulation by an extra 100 nanoseconds, you can see that both tails begin to enter the receptor. And also we have the it's continuing after the 100 nanoseconds to continue towards the inside of the receptor. Presumably, if we extended this, it would possibly find its way to the core. So this phospholipid, our data brings up a number of implications for the receptor and for class C, GABA-B is class C receptor. So the phospholipid is here and it's coordinated by a residue, at least in the TM3, and we also believe that ECL2 loop is working as kind of a lid to keep it in place inside the receptor. So in knowing that the ECL2 loop is important for the signal transduction of GABA-B, we believe that the phospholipid may actually be some type of sensor between this ECL2 loop and the 7-TM domain, because remember the G protein is being sensed here on the GABA-B2, or is being engaged here on the GABA-B2 receptor. Additionally to that, there's an implication in drug development for GABA-B because allosteric modulators for class C receptors in general have been believed to bind in the 7-TM core region where we see the phospholipid. So in order for that to occur, allosteric modulators would either need to displace the lipid or bind somewhere else on the receptor. So it'll be very interesting in the future to see more of what exactly is this phospholipid doing. I think we have some clues. It's clearly doing something to the activity of the receptor, as well as having some type of structural stability. The future directions will be really interesting that people go and look at. So with that, I would just like to acknowledge my lab, especially Yorgo Skiniotis for his mentorship, Mike Robertson, who is co-first author on the Nature paper that I'm discussing today, did the modeling as well as the molecular dynamics simulations. Uliana and Alpai helped with the cryo-EM data collection, and Shenhui helped with training me with cryo-EM. And Jesper Matheson at University of Copenhagen conducted the activity assays. And with that, I will take any questions.
Fantastic talk, Makaia. So I'll read out the questions for here. So John Janetsko, you had a question?
Oh, hi. Yeah, so I was curious in your toggle switch mutant, if you think that it gains the ability to recruit a second G protein when you add this.
Okay. Whether it would gain the ability to recruit a second G protein. So you're saying if it had the ability to bind GABA-B1 and GABA-B2. So with GABA-B, only the GABA-B2 receptor engages the G protein.
Yeah, but you say that you saw an increase in efficacy when you make the installation of the tryptophan.
Oh, I see what you're saying. I don't know. That's actually a very interesting point, because maybe that phospholipid is changing the way the TMs are able to move. Although I wouldn't think sterically, I don't know that you'd be able to bind because the 7-TM cores are closer together in GABA-B than they would be in, for example, in like GABA-B or in other class Cs. I don't know sterically if you could actually bind two G proteins at once. But whether it's a possibility that it can then engage, it's an interesting question.
Since other people had questions, I guess you could just answer the second part later and then someone else can ask a question.
Sounds good, John, thanks. I had a quick question before I give it over to John Burke. Did anyone look for sphingolipids instead of phospholipids? Because I'm sort of surprised that it's a phospholipid there's so many sphingolipids on the extracellular matrix. And I think in insect cells, sphingomyelin is not the major version. I think it's like a ceramide phosphoethanolamine. So it's just sort of a comment. You guys may want to like look at that, or if you guys looked at that, you could comment on that.
So we had thought about that, and we tried looking at the density. Based on our density, we thought it was phosphoethanolamine. Beyond that, I would say, like as I mentioned, there's another paper from King Fenn's group, actually, who they had done the mass spec, and in theirs they did find, they had 50% phosphoethanolamine and 50% phosphatidylcholine. So it seems like it is the phospholipid rather than the sphingolipid.
Okay, well, yeah, it may just be worth going back because the mass spec for phospholipids protocol's a little different for sphingolipids.
Oh, interesting. Okay.
So John Burke, you had a question? I don't want to take too much time.
Yeah, just a quick one for me. So from looking at the kind of kinks in the acyl chains on those lipids, the lipid has a pretty strong selectivity for specific unsaturation in the lipid tails.
Yes, it seems that way. We don't know for a fact, at least based on our EM densities. We didn't have high enough resolution that we could say for sure. But
okay, thanks.
Ray, you had a question?
Yeah. About the kinks in the acyl chains, you don't need to have an unsaturation in there to get a kink in the acyl chain, but yeah, that's a different thing. I was just wondering, and I guess you kind of suggested that you hadn't. Did you ever try to purify the receptor and then actually put in the other phospholipid? Like if you're finding it with PE, did you try to put in PC and try to determine if you could see a difference? You kind of suggested that you didn't do that because the other group did the mass spec, right?
Yes, and it's also, I think that would be interesting if you can actually force a different type of lipid in there and see what that would effect that have on the signaling of the receptor. I say that the lipid that we're getting in must be coming along. I believe it seems to me, based on the ability that we're able to get a decent density of the phospholipid, that the lipid is probably there in all the receptors for the most part. I didn't see any class of protein in which we didn't see the phospholipid, some type of density there. So it's probably being incorporated early on in the synthesis of the GPCR, I would think. Or as we saw in our simulations, there's a propensity for lipids to enter pretty rapidly. So whether or not you would have to, if we purified it, we would have to then displace it. And I think that would probably be different.
Yeah, and then the last thing is just if when you were fitting it, is it C16 that's fitting in there or C18?
C16. You mean with the kink or the?
Either or. Either. It was one of them too. It wasn't something.
So we didn't make a claim about the length because as far as the EM map goes, we couldn't say with absolute certainty.
So I think we have time for one last question. John Tesmer.
Hi, thank you. So this is an unusual arrangement for GPCR, as you stated. And based on your structure, do you have a mechanism for how ligand binding to one venous flytrap domain ends up allosterically turning on the transmembrane domain from the second chain, especially when there's so little contacts between them.
So we have, so I didn't present all the data in our paper, but the other structure we determined was actually a GABA-B1 homodimer, which adopts a similar conformation as far as the TMs are concerned, TM cores are concerned, as the mGlu5 active state, actually. And so part of the reason I bring this up is because the conformation change is actually that there's a TM interface change between, in our inactive state, we have a TM5 interface, and that changes to a TM6 interface upon activation. So you have that change. You also have, based on our structures, the cysteine-rich, not having a cysteine-rich domain, instead we have the extra long extracellular loop 2. The extracellular loop 2 in other class C receptors is actually interacts with the cysteine-rich domain. So it's probably a similar mechanism as far as that's concerned, getting to the VFTs to the 7TMs. Instead of using the cysteine-rich domain, it's sensed by the extracellular loop 2. I think the phospholipid is part of that sensing ability. How it gets to the other side, it may have to do with that inhibitory lock being broken with a change in TM3 that's involved in the inhibitory lock. That's our best guess so far, but there needs to be more work to be done to know what the precise mechanism is.
Thank you.
Well, fantastic talk, Makaia. There's a lot of questions that we can't get to, unfortunately, but maybe you can answer those in the chat box.
Okay.
So thank you. I'm going to hand it over to George Carman, who's going to introduce our next speaker, Joanna.
Thanks, Mike. It's a pleasure to introduce Joanna, also known as Asha Kwiatek, who was born in the United States, while her dad, who is Polish, was a doctoral student at the Brookhaven National Laboratory in Long Island. Subsequently, her parents returned to Poland, where Asha was raised and educated. She received her master's degree in biophysics from Jagiellonian University in Krakow, and Asha then began a worldwide adventure in science education, where she moved to Australia, where she received a PhD degree in biophysics at the University of New South Wales. Her doctoral work applied microscopy approaches to study lipid rafts in the plasma membrane. Following her stay in Australia, she moved to the United States to pursue postdoctoral studies in my laboratory. And as you will hear today, her research focuses on the enzymology and mode of action of the phosphatidic acid phosphatase enzyme from yeast. In addition to performing research, Asha is active with the Rutgers Postdoctoral Association. She is involved with the ASBMB, and most importantly, the chief organization and party planner in the Carman laboratory. Take it over, Asha.
Thank you very much, George, for a very kind introduction. I also would like to acknowledge John and Mike to giving me a chance to share my project with everyone who are listening to us around the world. It takes a couple of seconds to my computer to move to another slide, so my apologies for that, but sometimes too much technology is just killing me. Sorry, a couple more seconds. Oh, it's a bit bizarre. Okay, let's start then. The main actor of today's talk is phosphatidic acid phosphatase, to which I will be referring as PAP. PAP is the interfacial enzyme which dephosphorylates phosphatidic acid to produce diacylglycerol. The reaction, the PAP reaction, was for the first time discovered in 1957 in Kennedy lab. So since the entire timeline of PH1 started in 57, there were so many papers and milestone discoveries since that time. So as mentioned, PAP was discovered by Kennedy lab, and it happened in chicken. And for the next 30 years, many groups was trying to purify PAP enzyme from different organisms. As the first laboratory who managed to purify PAP was Carman lab and person Lee, who purified PAP from yeast cells. Coincidentally, Gordon lab identifies the fatty liver dystrophy mouse as having defects in lipid metabolism. For the next couple of years, Carman lab was trying to define the gene which stays behind PAP. Unfortunately, this work was not very successful, and the student Wu decided to drop this project, and she stored the purified PAP enzyme in the freezer. At the same year, Oshima lab identified SMP2 as a gene involved in plasmid maintenance and respiration. At the beginning of the new millennium, Rui lab identifies lipin-1 mutation responsible for lipodystrophic defects in FLD mouse. Importantly, lipin-1 is conserved in yeast. 4 years later, Sinosoglu lab identified SMP2 as transcription factor that couples phospholipid synthesis to nuclear membrane growth. 1 year later, postdoc Gilsu Han in Carman lab found purified PAP enzyme in the freezer, which was stored a couple years back, and established SMP2 as the gene encoding PAP. Nowadays, this gene is named PAH1. He also established human lipin-1 as PAP enzyme. Since 2006 towards nowadays, there were so many papers published about P phosphatase that would be impossible to list all of the discoveries. However, what is important that all of this research showed importance of PAP in lipid metabolism, which exemplified by cellular defects and lipid-based diseases associated with loss of PAP activity. Additionally, progress was made also on understanding the mode of action and mechanism of PAP regulation. And earlier this year, Arola lab published a paper in which they determined a crystal structure of the tetrahyamena PH2, which is also a PAP enzyme. So as I mentioned, PH1, which is PAP in yeast cells, is the enzyme which controls the conversion of PA to DAG. This reaction takes place in the nuclear ER membrane, and it's controlled by AHT-like catalytic domain, and it's magnesium dependent. PAP is also the main player in the regulation of lipid metabolism in cells because it controls whenever cells synthesize phospholipids in the exponential growth of or phase, or neutral lipid TG in the stationary growth phase. Additionally, PA, which is a substrate of this reaction, is a precursor for nucleotide CDPDG, from which all of the phospholipids are synthesized, and the product of the reaction is a precursor of TAG, which is stored in lipid droplets. Additionally, from DAG, phospholipids as a PC or PE might be synthesized via Kennedy pathway when choline and ethylamine are supplemented into cells. Many research published findings focusing on different phenotypes, stating that lack of PH1 in yeast cells cause major defects in lipid metabolism and cell physiology. The notable defects or phenotypes include reduced DAG and TAG levels, reduced lipid droplet numbers, elevated PA and membrane phospholipid vesicles, which leads to nuclear ER membrane expansion and derepression of phospholipid synthesis genes. Additionally, other phenotypes are also listed in this slide, showing the importance of PAP reaction. Also, we can mention elevated fatty acid levels and fatty acid-induced lipodystrophy, and sensitive to temperature. All of those phenotypes altogether lead to reduced chronological lifespan and programmed cell of death. In the current model of regulation of pH1 in yeast, PAP enzyme is phosphorylated by several kinases. This takes place in the cytoplasm, and phosphorylation retains pH1 in cytoplasm, which is far away from its substrate PA, which is present in the nuclear ER membrane. Additionally, this step attenuates PAP activity, stabilizes pH1, and recruits it to the membrane protein complex NEM1-SPOS7. Via this interaction, PAP enzyme is being dephosphorylated, hence it's getting translocated to the membrane, and its activity is stimulated, so it can perform PAP reaction. The dephosphorylation step also destabilizes pH1. Importantly, the unphosphorylated PAP, dephosphorylated PAP, or PKC phosphorylated, is degraded in protosomes. In yeast cells, pH1 is that's the name of PA phosphatase, and in mouse and humans, this enzyme is called lipin. In those enzymes, there are domains which are conserved, and they are called N-lip and C-lip, which are responsible for PAP activity. I would like to bring your attention towards the diagram, which shows different domains in yeast pH1. And starting from amphipathic helix, this region is responsible for membrane interaction. The tryptophan residue, which is located at the C-terminal within conserved sequence, is responsible for in vivo functions, and acetic tail is responsible for interaction with NEM1-SPOS7. Blue areas show intrinsically disordered regions, which are sites for phosphorylation. In our lab, based on biochemical studies, many of those sites were characterized, hence we know exactly which kinases are responsible for the phosphorylation. Because PAP is transiently phosphorylated at many different sites, that heterogeneity of phosphorylation hinders crystallization of full-length PAP. However, our Goro lab managed to obtain the crystal structure of N-lip and C-lip domain of tetrahymena pH2, which is PA phosphatase. And we could hear about this work at the beginning of this series. Many phenotypes were observed in mice and humans where lipin was not present or was overexpressed. Hence, in case of loss of lipin, we could distinguish lipodystrophy or neuropathy, and when lipin was overexpressed, obesity. Additionally, some of the phenotypes are also present in mice and humans, and among them there are insulin resistance and rhabdomyolysis. Hence, understanding the molecular mechanism which stays behind PAP enzyme is extremely important. Hence, in our project, we tested two hypotheses. The first one, the interaction of PAP with the membrane depends on phospholipid composition. And the second one, PAP activity depends on its ability to hop on the membrane, associate with its substrate PA, and scoot along it for additional Brown-Sud catalysis. In our experimental setup, we tried to mimic the nuclear ER membrane. Hence, we prepared liposome vesicle system, which also is called phospholipid vesicles. We used phospholipids, which are major phospholipids present in nuclear ER membrane of yeast cells. We used the physiological mole percent of those phospholipids. The average diameter size of liposomes was 100 nanometers. For PAP, It was expressed in E. coli, hence it was not phosphorylated, and it was bypassing NEM1-SPOS7. In our studies, we focused on investigating the interaction between PH1 and liposomes, and also we measured activity of PH1. Starting the interaction studies, we determined the amount of PH1 which is needed to observe that interactions. Hence, we incubated the various amount of PH1 with the saturated amount of liposomes, and liposomes were composed of the complex phospholipid mixtures. Liposomes sedimented during high-speed centrifuge, and we collected two fractions. Supernatant fraction, where the enzyme was free, and liposome fraction with bound enzyme. Both of those fractions were analyzed with immunoblot. We observed that with the increasing amount of PH1, liposomes got saturated with the enzyme, and so the signal from the liposome was much stronger, but also the signal in supernatant also highly increased, which indicated that enzyme couldn't bind anymore to liposomes. Owing that 90% of the enzyme bound to liposomes when we used 75 nanogram of the enzyme, we kept this amount for the further studies. We were wondering how the composition of liposomes may affect the interaction between the enzyme and the liposome. So considering condition A, where liposomes didn't have any PA, so the substrate of PH1, we observed that 57% of the signal was coming from liposomes, and a quite high fraction also was registered in supernatant. When we added 10% more of PA into liposome, which is our condition B, we also decided to change the composition, phospholipid composition of liposomes. We observed increasing PAP activity in liposomes containing PA. However, the individual phospholipid composition of liposomes didn't have a major effect on PAP binding. Going further, in the activity, we measure the activity. So we were checking how PAP activity depends on the amount and phospholipid composition of liposomes. We were determining the inorganic phosphate by calorimetric assay. Hence, we incubated PH1 with the various amount of liposomes containing 10% more PA. Every data point along the fitted line corresponds to a test tube with the increasing liposome amount and increasing PA mole. All of the data were fitted into Michaels-Menten fit, and what we observed that with this simple phospholipid mixture, the activity was low. However, with the more complex liposome mixture, the intensity of activity significantly increased. Owing that the ratio between Vmax and Km was relatively similar to all of those most complex liposome mixture, we decided to keep the mixture containing 5 phospholipids for the further studies because it's mimicking ER membrane quite well. Additionally, this experiment confirmed that PAP activity depends on the molar concentration of PA, which leads us to behavior how PA phosphatase may hop into the membrane. So undephosphorylated enzyme hops to the membrane, and either it interacts with phospholipid molecule or it hops to the membrane and it interacts with its substrate PA. We also determined that PA phosphatase activity is linearly dependent on time and on PH1 amount using complex liposome mixtures. Hence, PAP reaction follows zero-order kinetics. In many previous studies, micelle mixed system was used to mimic membranes. Hence, we compared the activity of liposome system versus mixed micelle system containing detergent and PA. We observed that with the very simple liposome mixture containing just PC and PA, the PAP activity significantly decreased in comparison to micelle activity. However, with the complex liposome system, the activity significantly increased in comparison to the activity observed in the micelle system, which also confirms that liposome system is more reliable and suitable to use it as a mimicking system for the membranes. Taking into account that PAP operates on the membrane, we decided to check how PAP activity is dependent on the curvature of the membrane, because membranes are not flat structures. Hence, we prepared 3 different sizes of liposomes, 50, 100, and 300 nanometers. We observed that PAP activity was significantly higher for liposomes with diameter of 100 and 300 in comparison to the smallest liposomes. Hence, indeed, this experiment indicates that PAP activity depends on the membrane curvature as well. Owing that around 50% of PA in yeast cells is composed of 60-81, that's the composition of fatty acid. We tested different compositions of PA. And also I should mention that routinely in those experiments we use 18-1-18-1 composition. This experiment didn't bring any major differences in phosphatidic acid activity for different composition of PA. Hence, we could conclude that really fatty acid composition didn't play a major role. Finally, we were wondering how PAP activity depends on the surface concentration of PA. In these studies, we incubated pH1 with the saturated amount of liposomes, where we varied the mole percent concentration of PA. It means that every data point along the fitted line corresponds to a test tube where liposome amount was the same, was constant. We only changed the PA mole percent, so the surface concentration. So those data were fitted into the Hill's equation, and the enzyme activity exhibited positive cooperative kinetics with respect to the surface concentration of PA. Whenever Hill number 2 reflects cooperative binding of 2 enzymes or 2 substrates, it's still unclear. However, previous studies showed that pH1 is monomeric. We favor a model by which the binding of one PA molecule facilitates interaction with second substrate molecule. Also, I would like to bring your attention to Km value, which here in our studies, we found it was 0.7 mole percent, which seems to be lower than physiological value. However, it might indicate that even the very small change in the PA mole percent in membranes may cause a major difference for PAP activity. That brings us to the model how PA phosphatase scoots along the membrane. When the enzyme binds to its substrate PA, it converts it to DHE, and then it scoots along the membrane to find another PA molecule, and so on. Taking into account all of our findings, we would like to propose the model how yeast PA phosphatase hops and scoots along the membrane. After PAP is being dephosphorylated, it hops to the membrane and it either binds to a molecule of phospholipid or it binds to its substrate PA and converts it to DHE. After that, it scoots along the membrane to find another PA molecule and to convert it to DEG, and so on. Some of you might find that the scooting mode might be favored. However, hopping mode might be also physiologically very relevant because some studies found PH1 in also different membranes, not only nuclear ER membrane. With that, I would like to thank my postdoctoral advisor, Dr. George Carman, and other members of my lab for lots of help. I would like to thank all of you for the attention, and I'm happy to take questions. Thank you very much.
Great talk, Joanna. We already got a couple questions, bunch of questions coming in. So, Tiago, you have a question?
Yeah, Joanna, really nice. And yeah, I'd like to know if you tested in your system the role of pH in phosphatidic acid phosphatase activity, and if you haven't tested, what would be your prediction in the activity?
So I must say that we haven't tested the pH of the membrane or of the liposomes. We used the optimal conditions in which pH1 operates and stays stable. So I'm wondering that if we change simply the pH, the enzyme could not be active. So that's why we just stick to this optimal conditions in which enzyme simply stays happy.
Yeah, the reason I'm asking this, and maybe we can talk about some other time, is that pH can also change the properties of phosphatidic acid, and it could also alter its availability. Thanks.
Thank you.
Laura, you had a question?
Yeah. Hi, Joanna. Really nice talk. So we talked a little bit about this before, I think, at the Gordon conference a while ago, but I'm just wondering if you accounted for charge in your complex liposome mixtures. So you're including phosphatidylinositol, so it would change the charge. So I'm wondering if that could account for this increase in activity.
Yeah, so what we observed that actually the activity was much higher with the more complex mixture. So this is definitely one of the predictions that it might play a role. Because indeed we saw that either when the mixture contained PCPE, PI, and PA, or PCPE, PSPA, then the activity was slightly higher than when it contained all of those 5 phospholipids. So yeah, it's probably I guess it does play a role, but as I mentioned, having all of those major phospholipids present in the membrane, we kept that to mimic as close as possible the nuclear ER membrane.
So I got a 2-year-old here, so we'll do Dan and then Amy after that.
Thanks. That was a very nice talk. I was just wondering, especially in light of the questions you just had, do you have any idea if the composition of the liposomes affects the residence time of the enzyme on the membranes? So does that sort of favor a scooting versus a hopping mode depending on the rest of the lipid composition?
So this would be definitely great to test, because we haven't tested any dynamic studies, and I think it would be very interesting to add this molecular dynamic to actually rule out this possibility, which enzyme composition is favors this interaction, and to keep the enzyme either longer or shorter on the membrane. But to be very honest, at the moment, I don't know the answer for your question, because also we haven't tested that at all.
Okay, thanks. It just may also change the rate at which it hops, and that you might be able to look at that, but thank you, that was good.
Yeah, thank you.
Amy, you want to ask your question?
Yes. Hi, Joanna. Nice talk. I also had the question about charge, so I'll go on to a second question. Is in vivo or by structure of the protein, are there hints that there's protein interactions that influence recruitment to specific site or retention at the membrane, the scaffold for the PAP?
So as far now as we know that PAP interacts with NEM1-SPOS7, and in our lab there are some biochemical studies happening to really determine like which region of this protein complex is responsible for PAP recruitment. However, still the crystal structure of PH1 is not discovered. So I think that at the moment we can only rely on biochemical studies for that.
Okay, and then there is a question from Vanina Zarenberg. Vanina, are you still there?
Yes. Hi, thank you very much. Very nice talk. I was wondering if you have tested the effect of increasing concentrations of DAG in PA containing liposomes.
We haven't done that. Yeah, we just was playing with the different phospholipid composition in liposomes.
Okay, thank you.
Okay, that's the end of the question. So I guess I will lead a virtual round of applause as everyone's muted. Those are two really great talks. Thanks everyone for coming. We're going to take a couple week break here, as I said, so keep an eye on your email for when we'll be starting again. Oh, we got one more that just popped up. Renok Dio, do you want to ask your question?
Late question, but I was just wondering if you delete is the PA interacting motif and the N-terminal hook of PAP the same? And like, if you delete the N-terminal region, do you see a decrease in activity?
So in for these studies, we didn't use any mutants, and this is something what definitely we want to look for the future studies. How the different parts of PH1 would interact with the phospholipid vesicles.
So the way PH1 interacts with phospholipids, it's not via the N-terminus, or is it different from the PA?
It's via amphiphilic helix, and some previous studies in our lab did the experiment where they used mutant, so where this amphiphilic helix was not present, and they checked that with the micelle system, as far as I remember. Yes, so that was confirmed that actually this amphiphilic helix like favors, of course, the interaction with the membrane.
Okay, all right. Thank you so much.
Yeah, thank you.
Great. Shamshad, you have your hand up in the virtual.
Yeah.
Question.
Can I ask a question?
Shoot.
Yes, that was a great talk. Now, my question is that if you had phosphatidic acid at the plasma membrane, would your pH protein attack that as well?
If I'm understanding properly, you're asking if the PA molecule is on the membrane, if the PH1 interacts with it. Is that correct?
No, at the plasma membrane, because you're looking at the yeast cells, and you said the protein PH1 is in the nuclear ER membranes. So the question is, why will it attack PA at the plasma membrane? And if not, why not?
Well, so the major place where PH1 operates is nuclear ER membrane. I cannot recall any papers which would state the presence of PH1 in the plasma membrane. I know that there are some reports showing that pH1 was localized even in lipid droplets or in vacuoles or the contact sites of ER. However, I can't recall anything about plasma membrane. Hence, I'm highly doubting if that's possible to localize pH1 there.
So the question is why, I mean, if it's the only molecule that's actually recognizing is the PA molecule, and PA will be present at the plasma membrane, what do you think is stopping the protein to attack PA at that location?
Well, so I think that all of the, so my understanding is that you'd like to show actually that if enough or if PA is present in the plasma membrane of course, that also the NEM1 SPO7 complex, which is responsible for the dephosphorylation of pH1, is located at the nuclear ER membrane. So that step recruits pH1 to the nuclear ER membrane, hence it can control the PAP reaction.
Okay. Thank you very much.
Yeah.
Okay, I think with that, we do have the end of the questions. So thanks again. And yeah, we'll see you guys in a couple of weeks. So keep an eye out on your emails for what the plan will be. Thanks a lot.